The overall goal of the following procedure is to use a high-throughput expression screening protocol to quantify in E. coli the level of soluble proteins using an automated liquid handling robot. This is accomplished by first inoculating 96 well pre-cultures with cells transformed with plasmids encoding the target fusion proteins. The next day, 24 well plates filled with autoinduction media are inoculated with the overnight pre-cultures to generate expression cultures. After harvest and freezing, soluble proteins from the expression cultures are then purified by immobilized metal affinity chromatography. Ultimately, the solubility levels for each intact target fusion can be measured to determine the optimal expression conditions for protein production and the generation of successful targets.
The main advantages of this technique over traditional methods are the increased efficiency, the limited batch-to-batch variation, and the simplicity of the data handling and tracking.
Visual demonstration of this method is critical. The automated steps can seem daunting and difficult to comprehend in text format alone, as the text does not convey the speediness or simplicity of the procedure.
After transformation of the bacteria, begin by using the robotic gripper to set aside the lid of the first 24-well LB agar plate. Next, use the 8-channel liquid handling arm to mix, then aspirate 50 microliters of transformation mix from a 96-well transformation plate. Dispense the transformation mix within